Good morning and welcome to day 2 of the 11th World Stem Cell Summit and RegMed Capital Conference. This room, as you know, is being live streamed, and most of our meeting is taped and will be broadcast and available. My name is Bernie Siegel. I'm the Executive Director of Genetics Policy Institute and Regenerative Medicine Foundation, which is the producer of this meeting. Of course, we are so honored to have our co-organizers, the Mayo Clinic, Kyoto University Institute of Integrated Cell Material Sciences, BioBridge Global, the New York Stem Cell Foundation, Wake Forest Institute of Regenerative Medicine, of course the Georgia Center for Regenerative Engineering and Medicine. Before I introduce our first of our two plenary keynote speakers this morning, I want to point out that this is a very busy day of the summit, and this is what we call Fantastic Friday. Tomorrow, as I told you, is Super Saturday, and all of you are going to change your flights and to be able to stay to the end because you want to take all of the Stem Cell Summit that you can in and take it home with you. Today, I want to point out there at noon, there are two tremendous symposia that are available to you. So if you've ordered a box lunch, you'll grab your box lunch and head to one of the two rooms, or you'll go down to the food court or the Starbucks downstairs. One is our annual, I think it's our 4th annual Japanese Symposium, Open Innovation for Large-Scale and Cost-Effective Cell Production Systems Using ES and IPS Cell Lines. Japan is leading the way with these technologies. We have representatives from Fujifilm and many of the leading Japanese countries there under the guidance of our friends Dr. Norio Nakasushi and Dr. Takashi Asada from ICEMS. This is a can't miss event if you're going to really keep up with what is going on in the world. And the second is we're very happy to have the National Institutes of Health in the house. NIH has a special symposium on funding resources for early stage regenerative medicine and life science companies. Free money. This is big news, and NIH is here to be able to provide funding for the research that's coming out of your labs that's going to be commercialized. You have to find your way to get this funding, CBER grants and such, and several of the institutes are here to help you do this. So if you're thinking about funding, if you're a startup, or just want to be able to take this back to your institution, to let them know, or to your board of directors to let you know there is a source of funding from our government, this is an opportunity to meet and interact with these people. Also this afternoon we have a special private workshop of the Center for Advancement of Science and Space that has an exhibit downstairs. So when you're ready to take your cells to the International Space Station, this is the place to be at the World Stem Cell Summit. CASES is also a platinum sponsor of today's event. There's also a special track keynote that I want to point out, 4 to 5. Our speaker, Ross Mason, who was unable to come, unfortunately, yesterday to give this presentation. It's the same time on the RegMed Capital track on venture philanthropy. If any of you know Ross Mason, he is a giant in venture philanthropy. Unfortunately, several years ago, he became paralyzed. He is a tremendous spokesman, a heroic individual, and that's just one of the concurrent sessions that are going on today. We have our exhibits and poster sessions at 5 o'clock, a great exhibit hall, and the posters are being reviewed by the poster committee, and we'll be announcing our top posters tomorrow morning. And there's also a special Fluidigm event, 7:30 to 10:30 across the street at White Oak Kitchen. Fluidigm always puts on a nice reception, and you should wander over there and also thank them for staging that, and make sure that you go by their booth today. Now, we have two speakers this morning. We have Dr. Joshua Hare from the University of Miami and Professor Tatsuya Shimizu, and I'm sorry, Professor, if I slightly mispronounced your name, from the Japanese Society for Regenerative Medicine. First, about Dr. Hare is a personal friend of mine. I'm a product of the University of Miami, undergraduate and law school, and I'm very proud to be on the governance board of the Interdisciplinary Stem Cell Institute of the University of Miami. that where Dr. Hare leads, that I'm very proud that his institute just received a $25 million gift from the Sofer Family Foundation that was announced yesterday. This is an institute that started at ground zero, that was pulling together resources from this great institution, and now is taking the stage as a world leader in this. Dr. Hare, of course, is an expert on cardiovascular, has running some major clinical trials, and has some exciting overview today about cell-based therapeutics as a broad spectrum. I will let him deliver this message in the best way and call him up today. So let's give Dr. Hare a warm greeting.
I wanted to pick a topic that was broad, and so my topic is cell-based therapeutics as a broad spectrum, new generation biological therapy. I have some disclosures to share. So this has been a very important field, a very new field, and a field that's full of controversy. But if we take a step back, I think we can see that we're learning some very important common themes across the cell types that we're trying to use, and starting to recognize the unbelievably broad applicability of cell therapy. And I think it's not unfair to draw an analogy between cell therapy in this century and antibiotic therapy in last century. So there are specific mechanisms of action that allow the therapeutic strategy to be very, very broad. So I'm going to cover these four general areas this morning: product manufacturing and availability, and allogeneic potential. So these are very much tied together because of the ability to use allogeneic cell products allows you to do product manufacturing and quality control at large scales, which adds a feature of practicality to the field. I'll talk very briefly about safety, and then spend most of my time talking about mechanism of action that leads us to contemplate this very broad disease activity. And in this regard, I'm going to talk about antifibrotic effects, immunomodulation, neovascularization, and pro-regenerative effects. So if we use the bone marrow-derived mesenchymal stem cell as a prototype for cell therapy, we can derive a schematic such as this for manufacturing steps. Now, the bone marrow-derived culture-expanded mesenchymal stem cell is a prototype, but there are many similar and related ways to produce cells that also yield large quantities of a cell product. So you could use a different tissue of origin, like umbilical cord, or you could add additional manufacturing steps, such as selecting a specific cell from the mixture based on a cell receptor. But this is the basic manufacturing overview of a bone marrow-derived mesenchymal stem cell. Let's see, do I have a pointer? Okay. That involves taking an aspiration of the tissue from a healthy donor, using a density centrifugation, obtaining the mononuclear cells, and then culture expanding them. Thanks, Keith. And then in this way, you can develop a production facility, a GMP production facility that can manufacture large quantities of these cells and therefore do clinical trials on a quality-controlled product. Now, again, as I mentioned, the immunology of these cells are fascinating, and I think the immunology really is the key here. It's the key to using cell therapy as allogeneic products, but it also has given our group, at least, and many other groups, great insights into mechanism of action. And I think it's this immunomodulation that is one of the central features of how these cells work, both in terms of immunomodulation, but also in enhancing of pro-regenerative effects. So as we learn more about the immunomodulation of mesenchymal cells and related cells, we recognize that it's very broad-based and balanced in such a way that it inhibits innate immunity, natural killer cell activity, dendritic cell activity, and adoptive immunity. This can be seen with a reduction in cytokine panels, but also by looking at T cell subsets. And here's some unpublished data from our laboratory where we looked at early and late T cell activation in a group of older individuals who didn't necessarily have any diseases. And you could see that they're both early and late T cell activation is suppressed by a single infusion of allogeneic mesenchymal stem cells. Now, what's very important about the cell effects is that they're very long lasting. And this is one of the central debates in the field, because we all recognize that if you give a single administration of a cell like a mesenchymal stem cell, that it washes out very quickly. And if you look at 3 months, there are no hints of presence of the cells. And this has been a big conundrum in the field, because how could these cells be having an effect, a regenerative effect, if they are washed out so quickly? But very clearly, we don't understand all of the facets of that, but they do have long-lasting effects. Wherever you look, whether you're looking at a pro-regenerative effect, an antifibrotic effect, or in this example, an effect on active T cells, you see that long-lasting suppression of the T cells. So just a quick word comparing autologous to allogeneic strategies. Essentially, the cell production is the same if you want to do a autologous mesenchymal stem cell product, but the only difference is that you have additional quality control steps that you build into when you use allogeneic cell therapy, and you yield a much larger cell product. We've actually had the opportunity to look at the safety of immunologic responses in a study called the Poseidon trial that was published in 2012. These were cardiac patients, and we did a detailed analysis of the PRA responses, the panel reactive antigen responses, to see if patients were tolerating allogeneic mesenchymal stem cell infusion. And without going through the slide in great detail, because the data is published, we find that cells are very well immunologically tolerated, even in a population like this that has a high baseline sensitivity. So with that, now moving on to safety profile of mesenchymal stem cells. I think one of the key drivers in this field that have allowed things to move into the number of clinical trials that have been done and for phase 3 trials to have initiated is a remarkable safety profile of mesenchymal stem cells. There are numerous trials now in numerous disease states, ranging from cardiac diseases to pulmonary diseases to pediatric diseases, graft versus host disease. The safety profile is really extraordinary for an early stage therapy. And in particular, there's an important publication in PLOS One in 2012 by Lulu et al., which is a meta-analysis of over 1,000 patients. Now, there's been a lot of safety concerns in the early stages of the field, particularly about immunologic side effects and tumor formation, but very nicely, all of these studies and analyses fail to reveal an increased risk of cancer in individuals who've been treated with mesenchymal stem cells. Okay, so now on to mechanism of action, and I listed 4 things that seem to be common features: antifibrotic effects, immunomodulatory effects, neovascularization, and pro-regenerative effects. Now, how can we tie all of these things together to understand what the cells are doing and to advance the field? So I think the antifibrotic effect is truly remarkable and a very, very prominent effect. It's also an effect that's without precedent in medicine. So if we shift the focus away from regeneration, which has been a very controversial area, but just focus on an antifibrotic effect, I think this alone leads us to recognize that we've got a new therapeutic modality on our hands. So these are early studies from our laboratory. These are pig hearts in which we've induced a myocardial infarction, which is a fibrotic scar, as you can see there, and it remodels the ventricle. This is the substrate for millions of cases of heart failure and sudden cardiac death worldwide. And this is the appearance of the heart after mesenchymal stem cell therapy. So there's a regression of the fibrosis, both in a total amount and circumferential extent, and therefore a preservation of the ventricular architecture. So we recognized that this effect was occurring over 10 years ago and have really used it in our studies both in animal models and in humans as a key endpoint. There is nothing, as I mentioned, there is nothing with precedent that can do this. There's no other therapy that could do this in our armamentarium. Now, if we go on to human studies, this is from the Poseidon study. This is a patient before and after therapy, 13 months after therapy, and you could see the remarkable regression of scar tissue in human populations. This is borne out very much in about 6 to 7 different studies that have been done that characterize tissue fibrosis. And if you look in this chart, you can see that the reduction in the MI size, which is the antifibrotic effect, is about 30 to 40% and consistent across all studies that have looked at it. So the antifibrotic effect has really driven the testing of mesenchymal stem cell therapy into a broader array of disease conditions. It was the basis for our hypothesis to do a study in idiopathic pulmonary fibrosis, for example. And as we can recognize, all of the chronic diseases that we confront that are incurable and with present medicine are diseases that have, in part, fibrosis associated with them. Now, what about the pro-regenerative effects? I think this is very important. And I think the common theme here is that cell therapy doesn't necessarily lead to an engraftment and differentiation of the exogenous cell that you administer, but rather a stimulation, a powerful stimulation of endogenous repair mechanisms. And as we've looked, we've seen more and more examples of this. So this is an early study published in 2010. The first author is Kostas Chatzigourgos, who's a postdoc in my lab. And he asked the question, do mesenchymal stem cells stimulate endogenous cardiac stem cells, cardiac CKIT cells, which are stained here in red in this depiction? And indeed, what we found was there was a massive upregulation of endogenous cardiac CKIT cells with the mesenchymal stem cell treatment. So there's a secondary amplifier effect, if you will. This is something I'm going to come back to and show you another example of in a study we did in humans. So it led to the idea that perhaps cell combination therapy could enhance a therapeutic response. If mesenchymal stem cells stimulate cardiac CKIT cells, why don't we mix them together and see if we can enhance the therapeutic response, jumpstart, if you will, or potentiate it. So we've actually had the opportunity to put that concept to test and are enrolling patients in a clinical trial right now. This is the animal data that supported that clinical trial. In these studies, we treated pigs with placebo, mesenchymal stem cells alone, or mesenchymal stem cells with culture-expanded CKIT cells, and looked at antifibrotic and pro-functional effects. So these are the antifibrotic effects. They're the same with or without the CKIT cells. But when we look at measures of myocardial function, like ejection fraction, we see an enhanced effect with the cell mixture. I don't have the data. This is published just a few weeks ago, and we do show an enhancement of cell cycle activity in the hearts of the combination-treated animals. This has allowed us to go on and through the NHLBI Cardiovascular Cell Therapy Research Network design and initiate enrollment in a study called the CONCERT CHF Trial, which specifically tests this notion of cell combination therapy. And as I said, we've already started to enroll patients in this trial, and this is the basic design. There'll be patients receiving the combination, either cell alone, mesenchymal stem cell or cardiac stem cell, versus a placebo group. Now, a critical step in being able to conduct this trial was the ability to produce the second cell type, the CKIT cell. I already showed you how we produce the mesenchymal stem cell, and this is a technology that's broadly available. But the CKIT cell was a little bit challenging, a little bit more challenging. We were able to develop the technique for the GMP environment, and this involves taking a heart biopsy, collagenase digesting the tissue, running it over a CD117 column, collecting those cells, and then taking them through further culture expansion. And with the refinement of this technique, we've been very successful in developing several 5 to 7 million cells from a single endomyocardial biopsy, as shown here, and therefore being able to conduct that trial of the cell mixture versus each cell constituent alone. Now, I mentioned earlier that I'd show you another example of how mesenchymal stem cells activate an endogenous precursor cell, and this has turned out to be a very insightful study for us as well. I think it shows the general theme that the pro-regenerative effects of cell therapy, and mesenchymal cell therapy, does involve a secondary amplifier effect. And so we were very interested in finding a second example that was also relevant to another key feature of the mechanism of action, neovascularization. So it's long been recognized that EPCs, or endothelial precursor cells, are markedly suppressed in a number of disease entities, particularly heart failure, but in a very broad number of cardiovascular and non-cardiovascular disease entities. So chronic illness is something that's associated with low EPCs, and this is of functional significance. Blood vessel function is impaired in these disease conditions, and that can be measured by measuring the dilatation ability of the brachial artery. So in our studies, we had the opportunity to look at this, and this hypothesis was generated by the CKIT observations. We were looking to see if mesenchymal stem cells activated another precursor population, and one that we could easily measure in humans, and this can be measured from the circulating blood. And lo and behold, we see that allogeneic mesenchymal stem cells normalize the EPC abundance in the circulation. But interestingly, and this is helping us with our insights in the field, autologous therapy did not. So whatever is impaired in the autologous bone marrow from the beginning in these ill patients remains impaired after culture expansion in such a way that this EPC stimulatory effect is absent. Now, the stimulation of the EPCs has functional significance, and as shown here, the increase in EPCs correlates with the increase in forearm-mediated vasodilation. So the increasing EPCs do have functional significance. So what I've tried to show you is how our understanding of mechanism of action has allowed us to understand the therapy better, to refine it. And I think what we're seeing in the field is, in parallel, an advancement of the field into phase 3, coupled with greater mechanistic understanding. And this is just a chart that shows that, at least for congestive heart failure, we now have a number of phase 3 trials that are being conducted, and there'll be several thousand patients enrolled in total in cell therapy for congestive heart failure. So although the field has had rocky starts and lots of controversies, at least we can say we'll have the answers from a phase 3 trial in this decade. Now, the broadness, our mechanistic understanding and our access to producing these high-quality mesenchymal stem cells and other cell types have led us to really broaden our capacities at the Stem Cell Institute at the University of Miami. And we'd like to say that we've got an alpha clinic, which was a concept that was originated in California through CIRM. So this just shows the number of disease areas that presently investigators at the University of Miami are working. And we have dubbed a UHealth comprehensive program for regenerative medicine as our alpha clinic arm of the Stem Cell Institute. And we're very proud that we're up to about 10 different disease areas as depicted here, including having an expanded access program. And we've sought over the years a single patient IND in a number of instances, particularly a spinal cord injury and a very rare condition called stiff person syndrome. But you could also see how we're really entering into some territories that are very unique and I think surprising to the field. For example, in the CNS area, we're about to conduct trials for Alzheimer's disease and treatment-resistant depression. And I'll talk more about that in some upcoming slides. One of the studies we've done and we've completed enrollment we're very excited about is a study of intravenous infusion of cells for a condition called aging frailty, which is a biologically recognized syndrome, one that's growing in importance, but one that has no approved therapy. And we think that this syndrome is driven in large part by inflammation and by the suppression of endogenous precursor cells. So the mechanism of action of mesenchymal stem cells make perfect sense as a treatment for aging frailty. And we've conducted a 45-patient study. We expect to have the data next year. and we've measured things like exercise capacity, muscle strength, coordination, quality of life, bone density, heart function, and inflammatory markers. So our basic concept is that we can take advantage of what we know about mesenchymal stem cells to treat diffuse diseases that are inflammatory mediated, and through intravenous infusions reach the organs of interest, basically through the homing mechanism of inflammation, so that areas that are inflamed, as characterized in red, can be targeted by the mesenchymal stem cells through their homing, and these cells can have broad effects on a multitude of organs, including blood vessels. Just a quick word about why we think that mesenchymal stem cells should be tested in neuropsychiatric illnesses relates to the fact that these conditions are associated with inflammation, and particularly CNS inflammation. And so we'll be beginning trials that look at treatment-resistant depression and Alzheimer's disease that'll use as surrogate endpoints broad markers of inflammation, but we'll also start to look at markers of efficacy. So basically, I think the field has really matured. We can see how it can touch human beings very, very broadly across the spectrum of illnesses, across different organ systems. And our sense of having worked in this field, conducting rigorous trials, is that this therapy is probably more potent than we're recognizing through the conduct of our trials. I'll just show you a couple of examples of patients of ours who've made it into the popular press. This is one of a participant of ours in the CHF Trial who really was bedridden with cardiac disease prior to participating in the trial, who now has a picture in the New York Times of him doing breakdancing. Another very important case for us, this was a single patient expanded access case, was an Olympic athlete who severed her spinal cord while training for the Olympics a few years ago, who we did a single patient IND for to treat her both with intrathecal and peripheral injections. So I'll wrap things up. I'll just some concluding remarks. Adult allergenic mesenchymal stem cells and related cell types have broad potential as a therapeutic. These cell types are powerfully antifibrotic, stimulate neoangiogenesis, and improve endothelial function. They're very safe and effective. We think that allogeneic cell therapy may have advantages over autologous therapy, and I've shown you that data. Promotion of regeneration is mediated through the stimulation of endogenous precursor cells. We'll be able to examine phase 3 data very soon, and we're already testing advanced concepts such as cell combination therapy. I need to end by thanking the terrific team of colleagues and trainees at the University of Miami and very important funding sources, the NIH, of course, but the Star Foundation, the SOFA family, and the Marcus Foundation all have been very valuable supporters of our work. Thank you very much.